Hi everyone, my name is Greg Sawyer. I'm a professor at the University of Florida, and today I want to talk with you about contact lens surfaces and the way that we're using these surfaces in biomimetics to try to increase lubricity and reduce cell damage in corneal epithelial cell models. Here's my disclosures. As we all know, the eye is a delicate system of two epithelial surfaces, the eyelid and the cornea, that experience sliding. And these surfaces are protected by the tear film, which is a lubricious gel-spanning network made up of a complex assembly of mucins. And as you see here in the right, it is this complex assembly of mucins that basically protect and separate the two epithelial surfaces. Inserting a contact lens could be a dramatic event because the contact lens is 15 to 20 times as thick as the natural tear film. But importantly, it actually creates two new interfaces. It takes a single biologic interface and separates it into a cell and synthetic surface interface on the eyelid side and a cell and synthetic interface on the corneal side. And our hypothesis has been that inflammation is the biophysical link between lubricity and comfort. And it's this lubricity of these interfaces between biology and the synthetic materials that we're trying to bring in biomimicry to try to reduce shear stresses and reduce inflammation. I've mentioned shear stress now on the first two slides, so I want to just take a split second and drop down to understand a little bit of the physics of what we're talking about. You've remembered in the past the discussions of friction coefficient, which is a ratio of a friction force, which we see here is that force to drag that lens across the surface, to the normal force, which is that pressure or that force that we're pushing down on that contact lens. Now, as that surface deforms, it deforms over a finite area, A. So the larger that contact area is, for the same friction force, the lower the shear stress. And that's because shear stress is a ratio of that friction force divided by that area in contact. And as we look here on the right in the bottom part of the graph, apoptosis is a measure of cell stress, cell damage. It's a natural cell death process, okay? And if we look at the x-axis where we have average shear stress in triplicates over 1,000 cycles, what we've done is we've caused of controlled shear stress sliding back and forth across these cell models. And what we can see is that as we go over 40 pascals of shear stress, we start to detect an increasing amount of apoptosis in these cell models. So it looks like the shear stress threshold is something that's important in terms of cell damage. And it's interesting that in this model, it looks like the physiologic level is the level that biology has basically designed itself to. It tries to stay below this 40 pascal level. The other thing that happens is with shear stress, and we mentioned inflammation earlier, is that as we increase that shear stress, we also increase the production of pro-inflammatory cytokines, such as IL-6 and MP9. Now, these cytokines are linked to pathological pain. And it may be through the cytokine production that our eyes are detecting pain. And we're detecting this as it accumulates both by increased shear stress and increased number of sliding cycles. So this is the deep dive that we're going to take. We're going to be using our tools in cell biology to understand the cellular response to measurements where we are going to probe how they respond to these lens surfaces under physiological levels of pressure, stress, sliding duration, and sliding speed. So let's talk a little bit about design. Traditional design with a homogeneous lens material is one of compromises and trade-offs. We know from polymer physics that increasing the water content increases the mesh size, increases the softness, and increases the lubricity of those gels, those aqueous gels. We also know from polymer science that if we increase the polymer concentration, which decreases the water content and reduces the mesh size, that increases the stiffness, the modulus of the lens. That increases the oxygen permeability because we have control of those polymer constituents. That eases handling, and that assists in vision stability. So in this trade-off, we have to trade these two things, the things that we want for lubricity with the things that we want for the control of the lens. The recognition and the breakthrough that we had was one of recognize this is a surface phenomenon. So why not put the high water content on the surface of the structure and the high polymer content in the core? And that's exactly what's led to the designs that you see here on the right, Varrophil-Canae and Delaphil-Canae, where really we've used surface science to put the high water content material where it needs to be for the biology, next to the cells. We've put the high polymer concentration where it needs to be for transport properties and handling in the core. And if we take a look at the design structure now, what it allows us to do is take the best of both attributes. Increase the water content, softness and lubricity at the surface, increase the modulus, oxygen permeability, ease of handling, and vision stability in the core. And this is really the key behind this. So now, to what extent does the physics describe how this process works? Well, in a traditional lens that you might see on the left, if we put something in contact with it, what we see is a contact area that responds to that surface with the pressure distribution that you see right here and a friction coefficient, okay? Under the same applied loads, if you have a surface gel layer, what happens is that increased softness spreads the area out, so the area comes up, the water drops the friction coefficient, so the friction force comes down, and the product of a reduced friction force and a larger area reduces the shear stress. And what I'd like you to take away is what we see on the right is that lower shear stress really is biology's understanding of lubricity. So how are we going to then interrogate these model systems to understand the role of lubricity back on cell damage and cell stress? What we end up doing is taking our cell models that I've shown you before in other years, but there's an image of it over here on the right, okay? What we have is a relatively large area of human corneal epithelial cells that are grown to confluence and are producing all of their mucins in monolayer. And now we're going to take the probe, as we showed on slide 3, and we're going to slide back and forth across that. And at different time points, we're going to stop and we're going to gently stain with propydium iodide, which is a validated nuclear stain that's going to indicate cell damage. And we'll see it in red, as you see right here. Then we use AI algorithms to basically track all of those cell borders of cells that are staining positive. We get the nucleus as well as the say border for that. And from that process right there, we get to stitch together a complete slide path analysis. And this is an example of one of those after 1,000 cycles of sliding. So now I'm going to show you the results of all of this work here in the following slide. What we see at the top is Delaphil-Canae and Varrophil-Canae. And we can detect 12 and 15 cells that are currently going through apoptosis. And these are the lenses that have the surface gel layers. Now, if we look down at Edephil-Canae, Stenphil-Canae, and Somaphil-Canae, we see a couple of trends. One, increased contact pressures. We can understand that now coming from the polymer physics of how the lenses are prepared, as well as increased friction coefficients. So the surface gel layers have lower contact pressures because they've spread that contact out over larger areas, and they have lower friction coefficients because they're higher water content. And the combination of those two things is leading to a reduction in cell stress and cell damage, as indicated here by the red. Now, what we do in order to make this quantitative is we use a cell counting method where we basically count these over areas. So now I'll report the number of damaged cells per square millimeter, as you see right here in this inset. Notice all these tests were run at the same normal load, 200 micronewtons, the same speed, 1 millimeter per second, and the same stroke length of 3 millimeters. Here we're going to see now the cell damage associated with these lenses over 1,000 sliding reciprocations in triplicate. And you basically see these in the different test colors. The averages, as we say, are 321, 155, 137, down to 14 and 12 for the Varrophil-Canae and the Delaphil-Canae. Now, interestingly, cells are always going through natural apoptosis. And the baseline level for apoptosis is 7. So we're really getting down into these physiologic levels as we tune these surface gel layers in order to do this. The other thing that I think is important is to what extent do we see this as something that can accumulate? So we know how damage accumulates in natural systems. And if we look at this for the lenses that we see up here without the surface gel layers, what we see is over a number of reciprocations, basically a monotonically increasing amount of damage over time. In contrast, what we see here for the ones with gel layers is certainly a measurable but slight increase over the normal apoptotic process, but it appears to be essentially flat as we look at different time points. So we think the mechanisms of this are really associated with the following things: that the shear stress drives all of the physiologic processes that are associated with the pro-inflammatory cytokine production as well as cell damage. And that lenses with the higher water content reduce the friction, reduce the modulus, and show the lowest levels of this. Both friction coefficient and the elastic modulus decrease with increasing water content. So this is the physics behind that process. And try to remember that this physiological understanding of lubricity is shear stress, and that's a combination of contact pressure and friction coefficient. And together with all of this work, this allows us a design paradigm in future directions to continue to try to develop lenses that have this high water content surfaces to try to make them more comfortable through lower friction coefficients, lower surface elastic modulus, and lower shear stress. So thank you all for your time.